VAIDS Vaccine-Acquired Immune Deficiency Syndrome. Article title: Innate Immune Suppression by SARS-CoV-2 mRNA Vaccinations. The Role of G-quadruplexes, exosomes, and microRNAs. pmc.ncbi.nlm.nih.gov/articles/pmc901253/pdf/main.pdf. Abstract. This paper argues that SARS-CoV-2 mRNA vaccines fundamentally alter innate immune signaling, particularly suppressing type 1 interferon pathways. The authors link these effects to disturbances in cancer surveillance, DNA repair, neurodegeneration, and viral reactivation. They highlight mechanisms involving G-quadruplexes, exosomes, and microRNAs, concluding that risks may outweigh benefits. Introduction. The authors review evidence suggesting that COVID-19 mRNA vaccines, unlike natural infection, impair innate immunity rather than mimic it. While traditional vaccines aim to trigger a benign infection response, mRNA vaccines employ modified nucleosides and lipid nanoparticles that extend spike protein expression. This design, though effective for antibody induction, may compromise long-term immune competence, type 1 interferons, and cancer surveillance. Type I interferons, IFNs, play a central role in antiviral defense and cancer suppression. They induce antiviral proteins, promote antigen presentation, and activate NK and CD8+ T cells, specifically IFN-alpha and downstream transcription factors IRF7 and IRF9, orchestrate apoptosis and tumor surveillance through trail signaling. Vaccination, the authors argue, suppresses IFN pathways and thereby impairs tumor control, potentially influencing cancers linked to BRCA1-2 dysfunction, such as breast, ovarian, and prostate cancers. Evidence from transcriptomic studies shows that vaccinated individuals lack the robust IFN upregulation seen in infected patients. This suppression, coupled with observed downregulation of STAT2, IRF7, and IRF9, may diminish the protective functions of BRCA1-2, increasing susceptibility to malignancies and even neurodegenerative diseases like Alzheimer's. Vaccine design considerations. mRNA vaccines were engineered with modifications to evade immune detection and enhance protein production. Key innovations include pseudouridine substitution, 1-methylpseudouridine, guanine methylated caps, optimized untranslated regions, and codon optimization to enrich GC content. These strategies prolong mRNA stability and spike production, but also bypass normal RNA quality control mechanisms. The reliance on lipid nanoparticles and synthetic cationic lipids further amplifies spike expression. However, the vaccine's parenteral administration favors IgG over IgA antibody responses, limiting mucosal immunity. Importantly, long-term persistence of vaccine mRNA in lymph nodes has been documented, raising concerns about chronic spike protein production, GC enrichment, and G-quadruplex structures. Codon optimization increased GC content in vaccine mRNAs to 53 to 61%, compared to approximately 36% in native viral RNA. This enrichment promotes formation of G-quadruplex, G4 structures, secondary RNA motifs linked to cancer biology, prion formation, and neurodegeneration. G4S interfere with mRNA regulation and protein translation, potentially dysregulating oncogenes and suppressor pathways. The authors caution that vaccine-derived G4 structures may act as decoys that distract microRNAs from their natural regulatory roles. This could impair tumor suppressors like P53 while enhancing oncogene expression. They cite a case study of lymphoma progression post-booster vaccination as possible clinical evidence. type Y IFNs in COVID-19. Clinical studies show that deficient type 1 interferon responses predict severe COVID-19 outcomes. Patients with autoantibodies to interferons or genetic variants affecting interferon signaling are at greater risk of critical illness. Conversely, natural infection strongly induces IFNs, whereas vaccines appear to suppress them, raising concerns about increased vulnerability during the immediate post-vaccination window. Epitranscriptomic methylation and translation. Normal cells rely on EM6A methylation and balanced cap-dependent/cap-independent translation. Vaccine mRNAs, however, enforce prolonged cap-dependent translation, consuming resources such as EIF4E and disrupting natural gene regulation. This shift may dysregulate M6A modifications, linking vaccination to oncogenesis and increased viral susceptibility. Exosomes and microRNAs. Exosomes, extracellular vesicles transporting proteins and RNAs, play crucial roles in intercellular communication. Vaccinated individuals have been shown to release spike protein-bearing exosomes, which persist in circulation and may traffic to distant tissues. Experimental work suggests that exosomal microRNAs MIR148A and MIR590, upregulated in spike-expressing cells, directly suppress IRF9 and USP33, crippling type 1 interferon signaling. The authors emphasize that this mechanism differs from natural infection and may explain neurological symptoms. Exosome trafficking along the vagus nerve could contribute to dysphagia, dysphonia, or dyspnea, conditions reported in vaccine adverse event systems. DNA repair and adaptive immunity. The paper also links spike protein to impaired DNA repair. In vitro studies show spike protein can enter the nucleus and interfere with BRCA1 and 53BP1 recruitment, both crucial for homologous recombination and non-homologous end joining. Such disruption may compromise lymphocyte diversity via impaired VDJ recombination and long-term cancer surveillance. Liu et al. 2021 found downregulation of genes involved in telomere maintenance, cell cycle control, and pol-Lorsch transcription after vaccination, consistent with impaired DNA repair and immune vulnerability. These findings raise concerns about long-term genomic stability. Pathological consequences proposed. The authors connect these molecular mechanisms to observed and potential morbidities. Cancer. Suppression of interferon regulatory factor pathways, and BRCA1/2 function. Neurodegeneration. G-quadruplexes. Spike-induced exosome trafficking, BRCA1 downregulation. Autoimmunity and chronic infections. Impaired interferon signaling leading to viral reactivation. Cardiovascular risks. mRNA dysregulation linked to myocarditis and thrombosis. Liver disease and hematologic disorders. Exosome-mediated systemic effects. VAERS and epidemiological observations. Citing VAERS data, the authors note signals for myocarditis, thrombocytopenia, Bell's palsy, and other conditions. While acknowledging causality is not definitively established, they argue that the consistency of mechanistic pathways with reported adverse events warrants serious concern. Discussion and conclusion. The central thesis is that mRNA vaccines represent an unprecedented intervention that suppresses type I interferons, disrupts RNA regulation, and introduces long-term risks not seen with natural infection. The authors argue that these risks, spanning cancer, neurodegeneration, impaired immunity, and DNA repair failure, call into question the net benefit of mass mRNA vaccination. They anticipate booster campaigns may amplify these problems, as repeated exposure could further entrench interferon suppression and exosome-mediated signaling disruptions. While recognizing the vaccine's role in reducing acute COVID-19 severity, They conclude that the balance of risks and benefits demands urgent reassessment.